Good morning. Thanks for joining us. This is our fourth briefing on policy issues related to technology transfer. I'm Matthew Stepp. I'm the Executive Director for the Center for Clean Energy Innovation think tank here in Washington, focused on energy technology development. First, I want to say we've been working with the House Technology Transfer Caucus to put on these briefings, and I want to thank the caucus co-chairs, Congressman Ben Ray Lujan and Gus Billy-Rockus, for their leadership on these issues, and even more importantly, their staff, Brian Crone and Jeremy Peterson, for taking on such a complicated issue. We obviously wouldn't be hosting these briefings without them. So really, I'm just going to give a very brief introduction, and then we're going to dive into this. So why are we here? You're probably confused. As my organization name suggests, I work in the climate and clean energy space, not health and biotechnology. But what both issues share in common is that desperate need to turn advancements in the lab into viable products in the marketplace. So whether we need breakthroughs from universities or national labs in solar power or wind or electric vehicles to make them cheaper, we need similar types of breakthroughs to address Alzheimer's, cancer, Ebola, or any of the long list of diseases and ailments that plague humanity. So turning kind of the breakthroughs that are in the lab into viable products in the marketplace is very difficult. And it's been a 30 or 40 year plus policy issue. It's fraught with funding and institutional barriers, and it's often considered one of the biggest challenges to innovation, which is why I have a particular interest in what the National Institutes of Health and NCATS are doing and why it's so important. We've done a lot of, the United States in particular has done a lot of policy and program experiments in technology transfer, and it looks like what NIH and NCATS is doing is actually working. And so it's these type of success stories that are really important as other agencies and universities and labs look to do for tech transfer what NIH is doing. So this panel is relatively simple. We're actually going to discuss what NCATS is, their lessons learned, potential success stories, and what this may mean for other agencies and other kind of big national needs that involve technology transfer and innovation. So moderating this panel is a good friend of mine, Joe Allen, who's really one of the leading experts in the country on technology transfer. He's the president of Allen & Associates, which focuses on technology management and related issues. And prior to that, he was the president of the National Technology Transfer Center, and he also served in the Senate Judiciary Committee with Senator Bayh, staffing the Bayh-Dole Act, which, as most of you all know, is probably one of the most consequential pieces of technology transfer legislation in the last 30 years. So with that, I'm going to hand it over to Joe. He's going to introduce the panelists, and we're going to dive into this. Basic ground rules. Each panelist is going to give kind of 7 to 10 minutes opening statements. Then we're going to get right into discussion. We're going to go right until 11:30, so we have plenty of time to answer all of your questions and get a good informal discussion going. So Joe, it's all yours.
So when someone introduces you as a leading expert, you can only go downhill from there. And actually, somebody asked on the panel how we came up with the idea of this. And the reason was because Lily Portilla, my friend, put your hand up, and I were riding on an airplane out to California, and we had 6 quality hours together. So we'd actually just done a briefing for the caucus. This is the 4th briefing that's been going on. And we hadn't had a chance to catch up, so when you have a flight to California, you have plenty of time there. And Lily was telling me about some of the things that NCATS was doing. And I said, that's a fantastic story. That would be a great idea for the caucus, because what the caucus is focused on, as the name implies, is technology transfer, but not just the theoretical part of it, but for billions and billions of dollars the American public is investing in public research and development, really making sure that those research results really get out to the public where they really can impact lives. And when we came up with the idea of this, and our friend Brian Crone, who's out here, Brian is a staff person there for the caucus. We actually went back and talked to Brian about it, and Matt, but we never could have imagined how timely this would be. Because right now, if you look at any newspaper or you turn on your television, there's nothing more pressing than the obvious need to develop treatments for the diseases ravaging the world. So that said, this is something that I think is a topic of not just academic interest, theoretical interest, but it really impacts real lives. We hear a lot about the miracle of modern science, but if you think about it, There's only a few countries in the world that even develop drugs. And there's a reason for that. If you get into drug development, I don't know if you ever watched Shark Tank, I love Shark Tank. If you went to Shark Tank and pitched drug development, here's what you'd be saying. Give me $2 to $5 billion, give me 14 years, and I got a 95% chance of failing. What do you think the investors would say?
Next.
And that's the reality. That's the real reality of what we're doing. It's a lot easier to copy drugs once they're made, which a lot of people in the world do. But the United States is leading the edge of developing new drugs, new treatments. But it could not be more high risk. This is not anything, if you have a faint heart, do not get into drug development. Having said that, when you think about it and really look at what's available right now, I really didn't realize this until I was looking at some information on something. Out of the thousands of diseases, there's only about 500 that actually have FDA-approved therapies available right now, which is daunting. But then when you look at rare and neglected diseases, you got about 6,500 rare and neglected diseases. Only 250 actually have treatments available. So when you think about that in terms of real lives of our friends and families, and even personally, what could be more devastating than having a disease like that, and then realizing there's really not much medical science can do, certainly not to cure you, and again, not even to alleviate the pain. So this is a clear area where there's a need for public-private partnerships. And it's also a great example of bipartisan cooperation. Because having looked at that and looking at what the American taxpayers is funding and investment, which again is leading the world and we're on the cutting edge of research, but again, unless that research is actually translated into new drugs and treatments, it's a paper somewhere, but it's not really impacting human lives. So in 2011, Congress got together and created the National Center for Advancing Translational Sciences, which is called NCATS. And NCATS is a different kind of federal organization. It's really designed to help close the gap between research and clinical trials, which again is where a lot of these promising therapies die. What NCATS is set up to do, and Dr. Austin will give us more details about this, they're really designed to bring together the best and brightest minds in the public and private sectors, people who are on the cutting edge of research, who actually have commercialization expertise, and to pull partnerships together, which again have the potential to not just get to do cutting edge research, not just get papers out and win awards and get federal grants, but most importantly, to come up with things that really can impact human lives, not just here, but around the world. We have a great panel today, and they're going to give you their perspectives about how these partnerships work, why they came into them, what they think they're getting out of them, and most importantly, what's the bottom line? How are we going to get this research translated from the research laboratory, either in the company or federal laboratories and universities, and actually get out where people can use them? We're going to use the same format that we used before, which Matt mentioned. I'm just going to introduce the panel by their titles. They're all going to do a brief statement. After that, we're going to have questions and discussion with the audience. We have a cinematographer here that we've had brought in at great expense. And she told me that even though we have a little microphone on the camera, what I'll do is I'll repeat your question just to make sure it's recorded. And she also promised that she would only film me from my good angle. We're not sure what angle that is, but she's got a couple hours here to work on it. So let me just introduce our panel real quick, and we'll go ahead and get started. We're really delighted to have Dr. Chris Austin, who actually is the NCATS director. And he actually changed his schedule to be with us here today. And he's got a big conference right after this. But again, we really appreciate your taking the time because you've got plenty of things to do. But obviously, this is something that's so important to him personally. He was willing to be with us. The next person we're going to will be speaking is Ron Bartek. Ron is the president of Friedrich's Ataxia Research Alliance. And he's going to give us the patient advocate perspective. The next person will be Dr. Steve Seiler. He's the CEO of ASREX, which is an innovative small business viewpoint. Then we'll have Dr. Bruce Trapnell, who's the Assistant Director of Adult Cystic Fibrosis Center at the Cincinnati Children's Hospital, which will give us the nonprofit organization perspective. And then Paul Kaplan, who's the Program Manager at Genzyme, who will give us the viewpoint of the established companies. So that's our program. We really are delighted you're here with us today. And I'll turn it over, and we'll get started with Dr. Austin.
So thanks, Joe. It's great to be here. Thanks for all of you coming. And yeah, this really is important to me personally and important to us for a number of reasons that I think will probably become obvious as we go through today. I did find it wonderful and fitting that this program started via a collaboration on an airplane between Joe and Lily, sort of fitting for what we're doing here today. And I also want to emphasize the thing that Joe said, that there are many, many illnesses which most people have never heard of. Many people have never heard of Ebola before a few months ago. And actually, the reason I have to run out right after this meeting is I have two meetings today on developing new therapeutics for Ebola. But there are many other illnesses like this, and they go by sometimes funny names. You'll hear about sickle cell disease and another disorder called pulmonary alveolar proteinosis. We also work on things like amyotrophic lateral sclerosis, or ALS, which is the subject of that ice bucket challenge. You remember that? But as a physician, I spent my career seeing these patients, and just imagine that there are thousands of these diseases that have no treatment. And as a physician, I was completely unable to treat any of these. And when I was in training about 30 years ago, we all sort of had peace with this because we didn't understand those diseases. But in the last 30 years, partially through the Genome Project and lots of other advances in science, we understand things that we simply didn't understand 30 years ago. And we have an ability to make new therapeutics that we simply didn't have 30 years ago. And so we feel very strongly that we have not only the opportunity, not only is there the need, but I would put it in moral terms. I think we have a moral obligation to the people out there who depend on us to deliver on the promise of science now that is possible. And really that was one of the main reasons why NCATS was formed 2.5 years ago. So what I often have to define, what translation is. I think everybody's familiar with, say, the National Cancer Institute. Everybody knows what cancer is, or the National Eye Institute. Everybody knows what the eye is. But what the National Institute for Advancing Translational Sciences, I've actually been asked by people on the Hill, so you guys do linguistics, right? So you do translational science. So translation is the term of art that's used for the process that you're all familiar with, which is the process of going from an observation in the lab, or in the clinic, or in the community, to an intervention, a drug, a device, a diagnostic, that has an improvement, that gives an improvement in the health of an individual and a community. And that whole process of creating the intervention and then deploying it in the community is what we term translation. But you'll notice that NCATS is not the National Center for Translation. There's lots of parts of NIH and the other parts of the ecosystem that do translation. We are the National Center for Advancing Translational Science. And it has been pointed out that we're the only institute in NIH that has a verb in the name, advancing. And that's really important to us. We are rate of change people. We are people who are fundamentally unsatisfied and impatient with the current way of doing things. And we measure everything we do to see whether what the new principles and paradigms that we're developing actually speed along the process so we get more treatments to more patients more quickly. I would say there's, you might say, well, hasn't this been an issue for a long time? It is an issue that's been a long time, but it's an issue which has even greater, importance than it ever has, given the kinds of opportunities that we've talked about. And you're all familiar with the really epical changes which have happened in science and medicine over the last 30 or 40 years. And if you ask yourself, have those fundamental changes in our understanding of how we work, and how human beings work in health and disease, have those logarithmic improvements been mirrored by logarithmic improvements in human health? The answer is absolutely not. And so why is that? One of the major reasons, and again, this is one of the reasons that NCATS was formed, is that we don't understand the general principles by which translation happens. And so that's what we mean by translational science. And translational science is the field of investigation that seeks to understand what are the general principles by which you go from an observation to an intervention that works in people. How do you make it less trial and error, in other words? So NCATS is, as you heard, it was formed to be a different beast in the research ecosystem. And we have a number of core principles, but I would say the biggest two of them are demonstrably useful deliverables, and you'll hear that today. And by demonstrably useful, I mean useful to somebody other than the person who created it. And frequently that is not just a publication, as I've been told by parents with kids with untreatable diseases. You know, I love publications, I love knowledge, but when my kid is sick, I can't give them a paper. And so we think about that every day. So deliverables and collaboration. Translation is an obligatorily team sport. I don't care how smart you are, I don't care how devoted you are, I don't care how skilled you are. This is, and I can use this analogy because of what happened on Monday night, this is football, and it really needs to be approached that way. Earlier in my career, I was in academia for a while, and I went to the pharmaceutical industry for a while. I was at Merck, and now I'm in the government, and we work very closely with all of those parts of the ecosystem. And another part of the ecosystem, which in my view has really been neglected up until now, and Ron will talk about this, which is the patient's point of view. We involve patients and patient groups in every project we do from the very beginning. And if you think about medicine as a consumer good, which really it is, the fact that the users of this technology that we're developing traditionally have not been involved in scoping out what those deliverables, what those products would be, is shocking, or silly, or stupid, or something. So the teams that we have are made up of scientists from the public sector, folks like Bruce from the private sector, that you'll hear from today as well, and from patient groups as well, and from NCATS. So what role does NCATS play here? We really think of ourselves as a catalyst. So what is a catalyst? A catalyst, biochemically, is vitamins are catalysts. Actually, they are entities which don't do anything by themselves. Actually, what they do is they bring together collaborators that otherwise wouldn't associate with each other, allow those collaborators or reactants to work together and create a product that they otherwise wouldn't. And so what you'll hear from today is folks who have tremendous expertise on their own, but for one reason or another, they were stuck. And we're unstickers, and we do that by these principles of teamwork, collaboration, and supplying the scientific entities, the scientific knowledge and expertise that's required to unstick these programs. So you'll hear about Trend today, so I just want to tell you about what that program is. We started this for the reason that Joe mentioned about 5 years ago, that though this is a problem across the translational spectrum, whether you're talking about Alzheimer's disease or diabetes, etc., but it is a particular issue in diseases which are rare or orphan, defined by the FDA as less than 200,000 people in the US, or neglected tropical disease, because the return on investment is traditionally lower for those. And so we formed this program, which had a very different model, like everything at NCATS, from how the rest of the ecosystem worked. That this isn't a program which gives out money. It's a program that says you have to have money to do these projects, and there are resources that NCATS brings to these. But the fundamental issue here is if you get into this program, you can put on a joint team, a joint project team, and that budget is given to the project team. And as long as the project meets its milestones and go no-go decisions, it keeps moving. And there's very intense project management, which Steve and Bruce, I'm sure, will mention. And these projects come in at the point where they're stuck. They've gotten to a sort of proof of principle point in the lab, but they're stuck getting through this so-called valley of death, which you'll hear talked about, getting to proof of concept in people. And generally, with these disorders, is if you can show that a new therapeutic has some indication of safety, so it's safe, and has some indication that it works in people, then you can attract a company, generally, who will come in and license that technology and complete its clinical development, do the large clinical trials that are required for FDA registration, marketing, et cetera, et cetera. So we take these projects just as far as they have to be de-risked enough in the vernacular I'm sure you're familiar with, to be able to fly on their own in the private sector. The last thing I'll mention, because it's a particularly important thing for us, is that we are innovators in everything we do, but we are innovators in both science, traditional science, chemistry, biology, computer science, engineering, but also in what you might call the soft sciences, if you want to call it that, the non-scientific, non-strictly scientific issues, which are responsible for about half of translational failure. If you ask why a translational project fails, just as often as it's a science, it's a collaboration issue, or an incentive issue, or an IP issue, or a business issue. So if those are issues which prevent translational success, and the purpose of NCATS is to increase translational success, then those are areas which have to be areas of innovation for NCATS as well. So we have a very robust effort in novel strategic alliances, which is the program that the office that Lily Portillo runs, to innovate in that space as well. And so and one of the things I'm hoping my co-panelists will talk about is what the arrangements were that allowed these teams to work, and what was really innovative about them. I am glad to tell you that what you're going to hear about today is tremendously gratifying for me, because they're successes, and they're proof of principle that this model works. And it's gratifying not only because we thought it would work, and we thought we knew what the secret sauce was, but until you could show this, we couldn't be satisfied, and I'd be sure that we were actually right. But you also hear about patients getting helped in this, through this as well. And what we're learning through each of those projects are principles, paradigms, scientific advances that we are applying to every subsequent project. So I'll look forward to taking your questions later. And who goes next? Ron, next?
The NCATS secret sauce really works, and it's really tasty. I'd like to begin by saying what a privilege, a genuine privilege and pleasure it is to follow Dr. Chris Austin. He's a tremendous leader in this space and a deeply appreciated colleague in this very important effort to develop treatments and cures for all the 30 million Americans with rare diseases. Speaking of which, I want to ask how many here either have a rare disease themselves, have a family member or a friend with a rare disease, or are an advocate of rare diseases? I think they're a good number, yeah. So any of you could step up here and say what I'm about to say, and that is that there's nothing that's preoccupied the patient or the patient advocate more over the last decade than the challenge of getting across the drug development valley of death to which Chris referred. I'd like to explain from the rare disease advocacy perspective how important NCATS has been and continues to be in getting us across that valley of death in three ways. First, I'd like to tell you a little bit about what patient advocacy organizations, like the several that are represented here in the room and ours included, try to get accomplished on their own with their own resources. And secondly, how NCATS and their NIH colleagues have been so wonderfully helpful in moving such organizations, some of which have resources, some of which entirely depend upon NIH and NCATS to move their ball forward, to become maybe become the 251st rare disease with a treatment. And third and finally, I'd like to tell you a little bit about my own personal experience, that of our own organization, the Free Drugs Ataxia Research Alliance, and working so well with our NCATS colleagues, and how NCATS has helped our own rare disease organization actually shrink the valley of death, make it narrower, and help us get across the other side. So first, what rare disease patient advocates try to accomplish on their own to get across the valley? First, we recognize that pharmaceutical entities and venture capitalists are far more interested in the rare disease space than they once were. Various reasons that probably most of you know, won't rehearse, but it's great that we now have additional interest and commitment from these very important partners. We recognize at the same time that they're not just going willy-nilly into the rare disease space and picking up, well, that sounds like a great disease. They're looking for what their CEOs call low-hanging fruit. And the task of the patient advocacy organization, therefore, is to grow low-hanging fruit. And what does that look like? It looks like, for the patient advocacy organizations like ours, the ability to assemble sufficient resources to at least begin assembling the scientific community, focusing the scientific community, and insisting on their collaboration with their, especially their academic investigator partners. It also means have the sufficient resources to begin funding the basic discovery science that is so important to attract the attention of a pharmaceutical partner, a venture capitalist, and other advocacy organizations. It begins too with when they take that next step down the path of the ability to develop some of the translational tools, not to translate Russian into English, but to translate discovery science into clinical science. And that could include things like cell models, assays, animal models in which new therapeutics can be tested to look for promising candidates for clinical research. It also means that they could actually begin to assemble a clinical network in which the clinical research can then be conducted by scientists, by clinicians and nurses and staff that actually know what a patient in that rare disease looks like and what their natural progression would be. And equally importantly, in such clinics, the advocacy organization can begin to assemble what's called a natural history database. What is the natural progression of this disease without intervention? Unfortunately, most of the 6,000 to 7,000 rare diseases have plenty of natural history because we don't have any interventions. But not many of them have those natural history databases so well organized that they can be a service to the pharmaceutical partner and help with all these approaches de-risk the program. So the venture capitalists, the pharmaceutical entities looking for patient advocacy organizations like ours to de-risk their investment, de-risk their clinical development, and knowing that the organization that has a patient registry that will be vital in recruiting clinical trials, because most clinical trials fail, and guess what? Most of them that fail because of insufficient patient participation. That will not happen to our disease, and won't happen to our other colleagues who have a patient registry that they use to identify patients that could be eligible for these clinical trials, and they're filled with eager patients ready to participate in developing their own treatments. And finally, the patient advocate tries to build the essential public-private partnerships with, first with their own patient population, building the trust with those patients so that they know that when that organization puts out the call for clinical research, they'll answer the call. Secondly, with the academic investigators, so important to developing treatments. The NIH and the FDA as our government partners, and industry, and even other nonprofits. So that's what the low-hanging fruit looks like. And as I said, some rare disease groups are large enough, sufficiently well-funded, that they can start developing some of that low-hanging fruit on their own. Even those, however, need the NIH and need NCATS to fill the gaps that they confront and don't have the resources, don't have the technology, don't have the expertise to fill. That situation confronted our little disease group, the Friedreich's Ataxia Research Alliance, this year when we already assembled a patient registry, a natural history database. We had a lot of basic research funded. But we knew that the long pole in the tent in developing treatments for us was biomarkers of our disease. Biomarkers are biochemical changes in the patient's cells that we can use to monitor progression and hopefully the potential impact, beneficial impact of a therapy in clinical trial. We don't have those biomarkers. We need them to advance additional candidates into clinical trials, especially in our younger patients that don't yet have full-blown symptoms of our disease that can't be used for those clinical outcome measures or clinical endpoints. So we want biomarkers that will show the FDA that there is potential improvement of these patients with this therapy moving downstream to clinical benefit. So we went to our friends and colleagues at the NCATS and talked with Chris Austin. He immediately agreed to help us facilitate a symposium, a workshop, if you will. We had 50 experts in technologies and science that were not available to us, outside of our disease space. Assembled on October 1st, just a month, less than a month ago. There at NCATS headquarters, Chris and his colleague John McHugh hosted the meeting, all-day meeting, 50 of the world's leading experts in the technologies and science that we needed to develop biomarkers in our disease. As a result of that wonderful day, we are already funding the highest priority biomarker projects identified by that group of 50. Some of them couldn't even spell Friedreich's ataxia probably, but they were certainly all over the kinds of technologies and the expertise that we needed to develop these biomarkers. And we are eternally grateful to Chris and his fine team for hosting that day. Let me go to the second scenario in which we know that most disease groups need NCATS help from the beginning. And not as fortunate as our disease group that had sufficient resources to do a lot of the basic science, get our patient registry natural history database together. You've heard Chris outline the NCATS programs that are so important to all of us as advocates. I'd like to emphasize just a couple. In addition to TREND and Bridges, that are mechanisms used to accelerate treatments for all kinds of rare diseases, and in a growing number, there's the Office of Rare Disease Research. That's one of the critical offices at NCATS. This office is working hard to develop, for example, a template that all rare disease groups can use to develop their patient registry and the natural history database. They're working very hard on that template. They're working in close coordination now with the National Organization for Rare Disorders, of which I'm the chairman of the board. They just launched the pilot project of a rare disease patient registry and natural history database for a single rare disease that then, after that's piloted, it'll be available to all rare diseases. And there's so many of those thousands of rare diseases that don't have the capability to develop their own patient registry and natural history database. This will give them the template and the process to be successful. Also, that Office of Rare Disease Research at NCATS runs and supports the Rare Disease Clinical Research Network. This is 22 consortia of as many as 8 to 10 sites each that conduct research, clinical research on at least 3 rare diseases. And they're stretched across the United States. They're working already with 98 separate patient advocacy organizations. And these are organizations that don't have the resources. This is our only opportunity to conduct live clinical research. And without the Office of Rare Disease Research and the congressional support for that network, just recently renewed, these patient organizations would be helpless and without any real opportunity to conduct clinical research, despite the fact they might have good basic science to translate. Finally, well, one other operation, a very important one there at NCATS, is the CTSA program. It's the Clinical and Translational Science Award program. It consists of 62 consortia around the country, our best universities and hospitals. Each consortium usually consists of 3 or 4 or 5 regional partners, and they're conducting research not just in rare diseases, but in common diseases. And there's no more critical need in clinical research than the networks that can pilot the clinical trials, that can conduct the clinical trials of these rare disease and common disease therapeutics. And there are no better networks available to us than the Rare Disease Clinical Research Network that NCATS supports and the CTSA program. Finally, I'd like to conclude with my own experience, that of our rare disease organization, the Free Drugs Ataxia Research Alliance, and that of the patients that we support and represent. I'm the co-founder and president of FARA, and here's our particular story of working so well with NCATS. First, we established FARA in 1998, a year after our then 11-year-old son Keith was diagnosed with free drugs ataxia. And we established the organization with a board of directors consisting of 7 patient parents like ourselves, my wife Rachel and I, and 1 adult patient, and the 3 leading scientists that we could convince they needed to devote their careers to the basic and clinical research for Friedreich's ataxia. Our first task was to assemble the field. As I said, we assembled the field in our first international scientific conference, in fact, the world's first international scientific conference dealing with this disorder in 1999. Only 65 scientists from around the world who could A, spell Friedreich's ataxia, and B, had done some experimental work in it, joined with their 15 colleagues there at the NIH. It was co-funded and co-hosted by our NIH colleagues, and that started our whole collaborative effort right from the get-go in 1999. There were no pharmaceutical entities willing. I called a lot of them. Many were called, none were willing to come, and they said, you're too rare, you're on your own. Then you fast forward to 2004, we met a little startup biotech. This is such a cool story. It's going to make me cry. So this little startup biotech comes to me in Washington, my little conference room at the business I was a partner in, and says, we've got 3 insights into Friedreich's ataxia that keep us awake at night. And so why don't we just treat this darn disease? I like the sound of that. I said, what you got? And he said, I've got 30 molecules I'd like to have tested against your best assays to identify a lead candidate. Who do I talk to? I introduced him to people at the NIH, people at our university scientists, the one that had our best assay for this mitochondrial agent, had our best mitochondrial assay. Got a call back 2 weeks later from that scientist saying, I've tested all 30 molecules. 3 of them are better than anything my assay has ever seen. 1 of them is 8 times better than anything my assay has ever seen. We need to get going on this project. We immediately decided that we would invest a large amount of our resources. We gave the largest research grant to that little startup biotech that we've ever given, $3.5 million in a research grant. We also invested $1 million in this biotech, both designed to get them on their feet. They're almost just coming out of being a virtual company, but they needed more help. So what did we do? We talked with our NIH colleagues. We learned about a brand new program that was part of Director Zerhouni's, the NIH Director Zerhouni's blueprint for biomedical research. and the roadmap for biomedical research. And it was called the RAID program, the Rapid Access to Interventional Development. It was designed for just what we needed, just what Chris Austin and his NCATS colleagues are trying to do today. Get a discovery through its preclinical hurdles and into the clinic. And it required, to be eligible for this program, a biotech or pharma, a PI, a principal investigator from an academic institution, and a patient advocacy organization. So that little biotech, the university investigator that had tested their molecules, and FARA became the three co-applicants and were the first non-cancer successful applicants to the RAID program. What did that do for us? It provided millions of dollars of resources into getting us through the preclinical hurdles, doing the toxicology studies in animals, doing the pharmacokinetics studies in animals, trying to decide where that drug would go and would it be safe. The net result of that was we got it ready for clinical research. It was in-licensed by a mid-sized pharmaceutical company that did the phase 1 and phase 2 clinical trials, both of which we recruited very promptly, and then went back to that little biotech that had then become a small pharma and was closing deals on investment and research that made them a larger and larger company. Cut to the chase, we finished dosing this month on the phase 2B, phase 3 of that molecule, for which we expect to have the data analysis done by the first quarter of this coming year. If those data look good enough, FARA and the company will go to the FDA, having already received fast-track status for that molecule. And if those data look good enough to the FDA to provide the basis for a new drug application, that drug could become our first approved treatment. So it's going to make me cry. But the importance of that is that we get that first approval. We'll go right back to the HHS Newborn Screening Committee, and with that approval, asked to be placed on newborn screening panels. We'll start diagnosing at birth, start treating infants, start delaying the symptoms of this disease, and give us more time for treatments 2 and 3 that will be profoundly beneficial. We already have 8 clinical trials going on right now that include gene-based therapies. We will have a gene therapy program itself into clinic within the next 18 months. So the cavalry has mounted. With this first treatment, we'll be able to get on newborn screening panels, and so the cavalry arrives at the infant's bedside. And none of that would be possible. So we have the potential. Our mission now is not just to treat and cure Friedreich's ataxia, but to eliminate it in the next generation. We're going to do that, and none of it would be possible without the help we've gotten from NCATS. Thank you.
Well, Ron, congratulations on your progress. So I have a neighbor with FA, and we're looking forward to the readout on your clinical program. Our drug, S103, is for the treatment of sickle cell disease. Sickle cell was first discovered in 1910 and was the first disease to be determined to be a genetic origin. That was Linus Pauling in 1949. And yet, in the over 100 years since sickle cell disease was discovered, there's never been a drug developed specifically to treat sickle cell. Now, this is a disease that afflicts 100,000 individuals in America and over 10 million individuals in Africa. This is a disease where, even with the best medical care, the life expectancy in the U.S. is 40 to 50 years. And in some parts of Africa, 80% of sickle cell patients never see their 5th birthday. With the help of TRIND and NCATS, S103 is now in phase 2 clinical trials. Without them, the 100-year-plus losing streak in sickle cell drugs might still be continuing. Now, let me tell you a little bit about our experience with S103 to illustrate why TRIND is important. When I first started working on the program, the sickle cell area was essentially moribund. Nothing was going on. In fact, when I went to my first sickle cell conference, there were 200 delegates, and I was amazed to discover that there was exactly one delegate, me, looking to develop a drug that was working in the private sector. S103 was also moribund. It was sitting on the shelf in a small, financially troubled biotech firm. And yet I thought it was one of the most exciting opportunities I'd seen. There was obviously a huge unmet medical need. And I also felt S103 was extremely attractive from a technical perspective. When I gained control of the program in 2009, I took it around to the usual suspects in the venture capital community, looking for funding to take the program forward. The response was generally very enthusiastic, actually. Broadly speaking, no one said no. They all said, come back with more data. Come back with human proof of concept. And why was that? Because the model we've used in the US for the translational development of drugs is broken. The translational development is the critical first stage of taking a drug from bench to bedside. That is the point at which the basic science is done. It's ready to leave the bench. But then there's an enormous amount of work required to turn that scientific insight into reality and to bring that to patients. This translational step has now become the place where drugs go to die. It's the place where all the brilliance of the basic research is wasted because nothing happens with it. It's the biotech valley of death. Now, the biotech valley of death is actually a fairly recent phenomenon as these things go. In the good old days in biotech, venture capitalists actually supported translational development. They then passed programs onto the public markets who supported clinical development. And for reasons I don't have time to go into in 7 minutes, the public markets now require a company to be in late-stage clinical development or even have an FDA-approved drugs to access them. So the public markets have moved downstream. This in turn has forced the the venture capital community to fund mid-stage clinical development. They too have moved downstream. It leaves no one but the NIH to fund translational research. This is exactly where SRX found itself in early 2010. An exciting program, an enormous unmet medical need, and no way to do translational development at S103. However, in early 2010, the National Heart and Lung Blood Institute, NHLBI, expressed interest in working with SRX as part of its intramural clinical program. Now, this was great news, but it was only one piece of the puzzle. S103 was still preclinical, and there was a lot of work necessary to be done before we could even engage NHLBI in the clinical development. And what's more, the NHLBI's program didn't give us enough data to provide human proof of concept. So we were missing an early piece and a late piece. In essence, we were still stuck. By the spring of 2010, Chris doesn't know this, it began to look as if this promising program would be in jeopardy. And I began to ask myself, is it time to throw in the towel? This is true. I'm going to cry. No. Maybe I will. When I met Trend, it became apparent that their mission was to fill exactly the gap that we faced. Moreover, our joint thesis was that if we took the program for where it was, to initial proof of concept, if we could bridge the translational gap, the program would be picked up on the other side by the more traditional sources of funding, the VCs, the big pharmas, who have the power to take it across the finish line into patients, which is what we're really talking about here, is getting stuff to patients. Trin became the essential catalyst in a program that ultimately involved not only SRX, Trin, and NHLBI, but also the National Institutes of Health's Clinical Center, the NIH Clinical Pharmacy, and the Bridges program at NCATS. We created a multi-stage, multi-institute, public-private collaboration research partnership. It's hard to say. It's even harder to run, as Chris knows. The collaboration also catalyzed external financing. After this was announced, we got another loan from the Massachusetts Life Sciences Center, and we were able to raise a couple $1 million from angel investors to keep our company alive. With Trin's help, we were able to complete preclinical development of S103 and get FDA approval to start clinical trials. We were also able to work with the FDA to define the endpoints that we could use for approval of a sickle cell drug. We undertook 2 early clinical trials, one in healthy volunteers and one in sickle cell patients. This one in sickle cell patients provided some really exciting data on the potential activity of our compound in sickle cell patients. And in fact, that data was presented last year at the prestigious annual convention of the American Society of Hematologists, or ASH. And we also started a longer-term clinical trial which would give us the human proof of concept necessary. S103 went from being nowhere to being the most advanced anti-sickling drug in development. And it's still a source of hope for the sickle cell community. Then in June of this year, we did something perhaps even more important. We made it to the other side of the biotech valley of death. SRX was bought by Baxter International, a Fortune 200 pharmaceutical company with the resources and expertise necessary to take S103 as far as its clinical success merits. Together, SRX and Trin have proven that our initial thesis was correct, that an appropriately de-risked molecule will be able to attract traditional funding on the other side of the biotech valley of death. So what we're saying is that this model works. We know it does, we've just proved it. And it works not just potentially for a sickle cell drug, but for any drug.
Not too good, so I'm going to use this stay close. Good morning. I'm grateful to have the opportunity to present an academic perspective in this forum. My name is Bruce Trapnell, and I'm a professor of medicine and pediatrics at the University of Cincinnati and director of the NIH-funded Rare Lung Diseases Clinical Research Consortium. I've studied rare diseases for 30 years with the goal of translating basic molecular research into new diagnostic tests and therapies in real time. Also, had an ardent supporter of patient advocacy and have been closely involved with multiple rare lung disease foundations over the years, including the Pulmonary Alveolar Prognosis Foundation, which I helped establish a few years ago, and which I currently serve as the scientific director. I'm going to give a little bit of background about the disease and the lung, so that this story will be a little bit more clear. And as background, the lung is composed of millions of tiny air sacs called alveoli, with very thin walls that allow oxygen in the air we breathe to pass through into the bloodstream. And surfactant is an oily substance made within alveoli, which forms a thin film that helps the alveoli stay open as we breathe. Used surfactant is removed by cells within alveoli that require stimulation by a molecule called GM-CSF to function normally and to keep surfactant from building up in alveoli. So that's the biology behind the disease. And autoimmune pulmonary alveolar prognosis, which I'll refer to as PAP, is a rare lung disease in which surfactant builds up in alveoli over time and eventually fills them completely. In a way, it's the opposite of what premature babies have. Their lungs are immature and can't make surfactant, so the lungs collapse. These guys have so much of it that they drown in it. So anyway, the excess surfactant prevents oxygen from passing into the blood and causes a low blood oxygen level, a feeling of breathlessness, and in severe cases, lung failure and death. PAP occurs in about 7 out of every 1 million people and affects between 2,000 to 3,000 people, men, women, and children in the United States. There's no specific therapy for PAP, and the excess surfactant must be removed physically through an invasive and inefficient procedure called whole lung lavage. In general, this is a procedure that requires general anesthesia and placement of a tube into each lung. One is connected to a breathing machine to keep the patient alive, and the other is completely filled up with salt water. And the chest is pounded to churn up the surfactant, which is oil, into the water. That's then drained out and repeatedly done for up to 50 liters per lung. And in several days, you look over to the side. That's the treatment. That's the standard treatment. It's not actually standardized, but that's the treatment. And I frequently describe this like washing butter out of a sponge using water from a garden hose. So it's a tough problem. research over the past 2 decades has lifted PAP from obscurity to clarity. The first clue was the identification that PAP develops in mice deficient in GM-CSF. The second clue was the observation of patients with then idiopathic or unknown PAP have an antibody, which is an immune defense molecule that's misdirected to attack GM-CSF. And our contribution here was to determine how GM-CSF deficiency caused PAP in mice. And then we proved that GM-CSF antibodies actually cause the disease in humans. We did a bypassive transfer of the antibodies from patients into animals. At the same time, we established methods to measure these antibodies in patient blood samples and then refine those assays into highly reliable diagnostic tests. And these simple blood tests now serve as the only means to identify this disease, autoimmune PAP. They also eliminate the need for lung biopsies, which reduces diagnostic morbidity. So finding out what caused PAP also led to a new therapeutic idea, which was GM-CSF augmentation. Fortuitously, GM-CSF had already been developed into a drug called leucine that was approved by the FDA for treating bone marrow diseases. And we had tried for a decade to establish a commercial partnership to evaluate leucine in PAP patients, but were unsuccessful. Notwithstanding, several small clinical trials were done and showed that it was safe and effective. These trials were largely done in Japan, very small trials. But unfortunately, these trials did not establish the effective dose or administration frequency or establish the length of time that would be needed to treat to have an adequate clean-out. So there was and this was important that these trials happened because the data from the trials prompted physicians to start prescribing leucine off-label, although inconsistently and without collecting any data or reporting any results. So it was impossible to learn from that. So because the clinical trials were needed, we applied to the FDA Orphan Drug Program for clinical trial support. And before they would review our grant, they required that we submit a new drug application, investigational new drug application, or IND. And so we complied with that. And they reviewed our IND, put it on full clinical hold, pending the completion of expensive formal safety studies. And our grant was actually never reviewed. And this was all very frustrating, especially since inhaled GM-CSF is actively being used to treat patients off-label, and again, not consistently with data collection. So sadly, we were unable to obtain funding for the required safety studies and were forced to discontinue our clinical trial plans, continue with our pathogenesis work, but we had to stop the trial plans. A year later, TREND was established as a mechanism to navigate the valley of death, which you've heard about. And just a reminder, that's that development area between publicly funded basic science and commercially funded clinical development, exactly where we hit the wall. We applied to the program, were accepted, and began planning required safety studies. One of the requirements was to identify potential commercial partners, and I indicated that Genzyme would be perfect since they focused to rare diseases and were very effective in that area, and currently we're producing leucine. Shortly thereafter, I received a call from someone at Genzyme, I think independently, and about possible interest in developing leucine for PAP. They had done their homework and figured that this was something that might be interesting. And I could barely contain my enthusiasm since the scientific rationale was so compelling. And we've been trying to do that for a decade. So after reviewing the data of the program, we entered into a three-way collaboration between academia, the government, and the pharmaceutical world. And since then, we've had bi-weekly conference calls. We've completed several of these required safety studies. And tomorrow, we return to the FDA with our TREND and Genzyme collaborators to discuss the next steps in our clinical development program. So in summary, TREND rescued us, our program, to develop the first pharmacologic therapy for PAP. So thank you, and thanks for TREND.
Good morning. First, I'd like to thank the organizers for inviting me to participate in this forum. My name is Paul Kaplan. I'm Senior Director of Program Management at Genzyme Corporation, Santa Fe Company. My current responsibilities include shepherding a development program for obtaining clearance to allow the use of Glucine for PAP, as Dr. Trepnell has just described. And this project would not have been initiated were it not for the funding from the TREND program. As Dr. Trepnell has just discussed, PAP is a serious and often life-threatening disease. It affects a relatively limited number of patients worldwide, considerably lower than the hurdle for orphan diseases in the United States, 3,000 in the U.S., and the orphan bar is about 200,000, so about 1% of that. Not only does it affect the individuals at a personal level, but it has a significant impact economically. We've estimated that it costs each patient, or costs the system, about $100,000 per patient per year in both direct medical costs and lost productivity. So it's a significant economic impact and a significant health impact. Nevertheless, our involvement with this program is a fortuitous story because I don't think this program would have gotten started internally at Genzyme were it not for this collaboration. Now, as you know, in order for a pharmaceutical company to market a drug, we have to have an approval from the FDA. We can't talk about marketing drugs off-label. That's illegal. We don't do it. But nevertheless, we are interested in looking at how we can use drugs to treat diseases for which there is currently insufficient data. And one of the problems with Glucine and pulmonary alveolar proteinosis is that there's certainly a lot of compelling scientific data that this could be an effective therapy. But that's a far cry from being able to go to regulators in the United States or anywhere for that matter and provide the data which shows how to deliver, what dose is needed, how frequently the drug should be administered, what conditions or what situations you wouldn't want to administer the drug. In short, we want to be able to provide physicians with adequate guidance to be able to prescribe a drug so that they can know that they're treating a patient effectively and they're not harming a patient. Certainly, one of the key principles of medicine is do no harm. And it is partly our job to provide evidence of efficacy, the proper way to use a drug, but also to provide sufficient data so that we don't run into safety problems along the way. Not only is regulatory approval required simply to market the drug, but in the United States, in most cases, regulatory approval is also required for insurance reimbursement. In an increasingly constrained environment, insurers will not reimburse for off-label usage of drugs. And if patients are to have access to therapies, there must be some mechanism to provide that drug, and that requires both the regulatory approval and insurance approval. Now, as Dr. Trepnell has mentioned, he's recognized the requirement for this scientific data, and he certainly applied to the FDA several years ago and was blocked in doing so. There was clearly insufficient animal data to justify continued development of this drug. And he was successful in obtaining NCATS funding to do this. Now, our involvement, Genzyme's involvement in this program was somewhat fortuitous. Genzyme is actually the 6th owner of the Glucine product over the last 20 years. And that may constitute a record for, shall we say, punting a drug from owner to owner. When we acquired the drug, it was placed into our oncology portfolio. Now, why oncology? This is a drug that's required, that is approved to mobilize certain cells in the immune system. Those cells are often damaged in certain kinds of cancer, or they are ablated in treating certain kinds of cancer. And hence, the approvals are all in the oncology space. And this drug went into our oncology portfolio. And that might well have been the end of this story, except for fortuitous timing and the appearance of some data. We had a scientist that Dr. Trepnell mentioned who was interested in pulmonary medicine. He had expertise in pulmonary biology, and he became aware at that time that Genzyme had acquired this product. We were a large company. Not everybody knows everything that's going on, despite what we'd like to think. And he called Dr. Trapnell out of the blue, as I understand it, and suggested a collaboration. And that timing was very fortuitous because that was just about the time he was obtaining the trend funding. And so with that, we put together this three-way collaboration. And one of the reasons that this worked with Genzyme, and it probably didn't work with other companies, is the fact that Genzyme certainly has an expertise in rare disease. We're a leader in developing rare disease treatments. I hate to say the smaller the better, but it might be true. This was an opportunity to move this drug outside of the oncology portfolio. And so I think a really great collaboration was born. But even there, why wouldn't we just pursue this? And again, it comes back to the valley of death. We can't just pursue every project that comes along. We have many projects. We work with many rare disease organizations. We have a limited amount, perhaps a fairly large, but still limited amount of development resources. And when we look at individual projects, we have to balance many things. We have to balance the expected timing of the development program, the cost of the development program, the maturity of the scientific data, the maturity of the patient data. As Ron has pointed out, you need to understand something about the natural history of the disease before you can treat it, because you don't know what your intervention is going to do. And so we have to balance all of those things as we put together a development program. And when we began to consider the merits of the Leukein program for PAP, it was clear that we weren't going to get enough resources in a timely manner to be able to do this entirely as an internal program. And that could very well have been the death knell for developing the program. I'm sure Dr. Trapnell would have continued that. But we certainly would not have. And that would have certainly delayed the program for a considerable amount of time. And so this funding from NCATS has allowed us to move forward at the speed that the data allows us to move forward. And that's really what drug development is all about. It's all about data. You want to make sure that you have the data that you need to provide efficacy, information, and the data that tells you when it's appropriate to use it and when it's appropriate not to use the drug. And so this has really turned into a win-win situation all around. It's certainly a win for Dr. Trapnell's research into both the cause of and the treatments for PAP. It's a win in that I think this is federal money well spent. This is a program where the data is out there telling us that it is very likely going to be a successful program. Forget the 95% failure. I think we're probably batting 1-1 on this one, if not more. It's going to be a win for Genzyme in that we can continue on our mission of developing products for significant unmet medical need. And I think most importantly that this is going to be a win for the patients because they're going to be able to have access to a drug which is very likely to work, and they're going to be able to benefit from all of the necessary regulatory and insurance information that's going to allow them to have access to that drug. Not simply know that it's out there and be unable to get it. Thank you.
I just want to start by making an observation. My friend John Preston ran the MIT Tech Transfer Office, and I used to bring him to Washington to talk to some of our political appointees, and John made the point, the single most important factor in commercialization is passion, and I think you can hear from the members of this panel that lack of passion is certainly not a problem here. But having said that, since we're talking to the Tech Transfer Caucus, one of the key factors in getting something from the lab to the market is intellectual property, particularly in an area as high risk and expensive as drug development. So let me just throw this out to the members of the panel. How do you manage patents that are brought into these collaborations? How are they shared with the other members of the alliance? And also, if somebody makes an invention under one of these agreements, how is it owned and managed? And anyone that wants to jump in on that, please.
Yeah, I'll answer that. So in all of the programs that we run, the so-called background IP that the collaborator has is theirs, and we want them to keep that, and that we're not involved in that at all. Sometimes there is IP that's joined, that's developed in the joint project. In that case, it's expected to be a real issue on a lot of projects. I don't think it actually has, because of those principles that have actually worked very well.
I can just provide maybe an academic perspective. I understand the importance of IP and that it has to be retained and managed. But a very personal, and this is just very limited personal view, I don't care about any of the money or the IP. I just want it to work. And we've dedicated the last 15 years to studying this, to understand it well enough, to get it to a point, and then to be stopped when it's so clear that it should be continued and pursued, to be stopped by anything really is a problem. And having a governmental organization specifically focused to making things work that doesn't keep the value necessarily, but just makes it work and go forward. It's just tremendous. So that's my perspective.
If I could add something about tech transfer, I agree with Chris that the basic intellectual property in terms of patents is generally covered because we come with foundation patents. We were a pilot program at Trin, and so we knew that part of the deal was that we're going to hit some potholes and we're going to have to work through them, and that was the handshake. So one of our potholes was that the actual data, and we're not talking about patentable information, but the underlying data is still owned by the federal government. And who has the use of that data going forward? Is it exclusive? Can anybody get it under FOIA? And I think we have some possible solutions. But that was a discovery that nobody would imagine would keep across. And it became a big deal with Baxter, a really big deal. We got there. Lily was instrumental in getting us across the finish line. But in retrospect, I think next time we would take care of that detail up front.
So, I'm not a lawyer, and I certainly don't represent the entire industry, but I think Genzyme in particular has a track record of focusing on patients, probably one of the earliest of the large pharma or large biotech companies to do so. And I think generally we've operated by the principle of do the right thing. We need to focus on getting the products out there, and we will work out an equitable situation. I think that the NCATS collaboration has allowed us to do so.
I can just add patient advocacy perspective. There are a lot of different approaches in the patient advocacy community. Our particular one is that we chose early on and stick with it. We do not hold any IP. It would take too large a staff, legal and otherwise, to manage IP issues. So we leave IP to the discovery scientists and the company that picks that discovery up. That's all up to them. The data that we collect in our patient registry and in our natural history database belongs to the organization and to the principal investigators involved. And those, the overall principal investigator of the natural history database meets with drug companies all the time, along with our staff. And we, with the expertise at the table, mine those data, if you will, to design clinical trials, to come up with biomarkers and clinical outcome measures to monitor during clinical trials. So, and companies can gain access through our key opinion leaders to those data in analysis sets, but not direct access to the data itself.
So any questions submitted by the audience? Yes, sir.
Any other organizations that seek commercialization and foster it in other fields, if I've understood you correctly, faces the challenge of bringing the risk of proof of concept research down at least sufficiently to where the private sector can step in. So how do you decide where to go and how selecting those discoveries for doing this? And two, when do you decide that the risk is then enough for the private sector?
So the question was, the NCATS is trying to de-risk some of these things, so how do you decide when and where to go and how you got the risk down enough the private sector can pick it up?
You just described the two hardest things we do. So how we choose projects. So two ways. One is we have a peer review group of about about 25 people who are from the biotech, pharma, venture capital communities, as well as academics who know about the translational process. And all the programs are selected through a peer review process. We also have about 150 people who do the work, a lot of the lab work too, at NCATS. About 80% of whom come from the private, have worked in the private sector, so they're familiar with both the science and the operational principles and the IP issues through this space. The peer reviewers are asked to assess the scientific state of the project, its likelihood of succeeding and getting to the other side of the biotech valley of death. A little bit, we ask them to evaluate whether they think there's any chance that if we did get to the other side, that there would be a partner willing to pick these up. And we look at whether there's any current treatment for the program. We have actually had excellent, for the disease, we have actually had excellent projects come in for a number of rare diseases that have actually gotten turned down because there are treatments already available. They may not be good, but if the alternative, we have many other projects which have no treatment at all, we tend to pick those. I should say the need is absolutely huge, and with the success of the program, we now have a truly heartbreaking process in choosing projects. The current number is 4% succeed in getting into the program. That's 4. But only 4, not 4%. So we turn down 96% of them. As far as how they get out, we do a tremendous amount of market research of the sort that any company would do. We're constantly out probing the community, the biotech community, we have a lot of connections with. But we also do what companies tend to do. We had, for instance, an R&D day in Cambridge, Mass. Last year, where we had our collaborators and the trend people and the NCATS folks present all of these projects in terms of the science, the potential commercial application, all of that stuff. We had term sheets that were available. Lily and her folks did all of this. And out of that came a number of leads that then led to a number of these projects graduating.
Any other questions? Or let me throw it out.
Let me just add to Chris's that in terms of the success, when I sat down with Chris in the summer of 2010, to your second question, when do we know when it's ready? We said the window will open when we finish our first trial on sickle cell patients. And the other side of the window is when we finish the bigger trial that we're undertaking. And we got a car by Baxter right between those two goals. So there is some success that proves that what they're doing is going to work.
So in addition to their technical expertise, when you're creating one of the 4% that gets funded, when you actually start forming those together, what are you looking for in a partner in addition to their technical expertise?
We try to assess this may sound funny, in addition to the technical expertise, and that means not only the scientific expertise, but the IP position too. That's really important for a partner to be available at the other side of the valley of death. But we also try to assess their interest in teamwork. And that can be very hard to assess. But frankly, it's the kind of thing that any coach would do. If you could have the best player on the field, but he hogs the ball and doesn't want to play on the team's going to lose. And so we have each of the potential applicants come in and spend a whole day with us, presenting their projects. We talk to them about what a potential plan would be, give them hypothetical scenarios. If this happens, what do you think we should do? What do you think we should do in this case? And get a sense of whether they're committed to the same direction we are. I must say sometimes there are folks who I know this will shock you, folks who have ulterior motives, who really are not interested in developing something for a rare disease. They actually are interested in developing it for something else, and they want to use our program to get to the something else. And so we try to ferret that out as well. We're actually trying to figure out how to quantify this. We were talking about it actually before today. How do you assess the quality of a collaborator first? Because we've actually found when our projects fail, they actually fail more often for these reasons than for science reasons.
couple times in your public statements that you're looking to go beyond just translational science and get into translational partnerships. So can you just say a little bit about what you have in mind, and how you really idealize to see this program move forward, and perhaps there being a model for other areas outside of diseases?
Yeah, I would say the biggest thing that we do is we, our starting assumption and attitude is that this isn't all about us, and we can't do this alone. And it really has to be a joint team which works together. It may sound odd, but there are rules, statutes, laws, and policies which actually stand in the way of our doing that, of actually sharing risk and cost with partners. And so we've had to be quite creative in coming up with new collaborative mechanisms. Some of this, most of it up until this point, have been policy issues at the NIH. Scientists, as you probably know, are fiercely independent, and I think they can do everything on their own. You can't do that in this space. And so Lily Portilla has been very creative through most often things called cooperative research and development agreements, which is the standard template that we use. But we've had to train the NIH people that the way we do grade is the way everybody else does grade is are fundamentally different. And then we put all of those, we put as much of those template agreements on our website. So we redact the business specific stuff, of course, but then we put agreements on our website that show people how we did it.
Reinforce it. When I gave my presentation, you might recall, I talked a lot about the kind of public-private partnership we were able to build in collaboration with the NIH that included the very mechanism, very much like a cooperative agreement that Chris and Lily just described. We had a pharma partner, the company, the little biotech. We had an academic principal investigator, and we had a patient advocacy organization. That was the kind of partnership we needed to advance. I told you about our use of what was called the Rapid Access for Intervention Development Rate Program. That rate program is now called Bridges. So that's one of the mechanisms that NCATS tries to use to build those kind of partnerships and that kind of team approach, which has worked so marvelously well for our group.
Yes, ma'am.
My name is Dr. Sasha, policy advisor for Congress and Housing Business of Florida. Interesting question, kind of atypical here on the Hill, and a neuroscientist by training. One of the questions kind of pertains to NCATS mission. One of the questions had to do with the demonstrative deliverables as well as collaboration. But it seems as though, this is not a criticism by any means, I love NCATS, but it seems to be a lot of the research goes more so toward therapeutics. Are there any initiatives that are aimed at looking at early diagnosis and new biomarker testing, especially microfluidic devices?
So the question, as I was able to hear it, was, in addition to looking for just treatments, are you looking for early detection, biomarkers, things that are sort of before you actually have the thing become full-blown?
Yeah, we're only talking about mainly trended bridges today, but many of the other programs, both the Rare Disease Clinical Research Network that Ron mentioned, as well as a lot of the activity in the CTSA program and other programs that we have, are focused not only on biomarkers and diagnostics, but also issues of innovative ways to do clinical trials and on not just therapeutics or even diagnostics, but devices and behavioral interventions, because we know that those are actually, in many cases, more effective than a drug.
And
But
what
we just didn't talk.
percentage of funding actually goes towards the deliverables, such as the therapeutics versus the devices?
Sorry.
Is there a breakdown in terms of the budget?
I don't think we've ever done that. But just to give you a sense of how, so the whole NCATS budget is about $670 million a year. The programs that we're talking about today, all of the productivity that you've heard about, total budget for that part of the program is about $30 million a year. So it is incredibly effective, and now we're applying a lot of those principles to the other parts of the organization.
We promised to get you out on time. What I'd like to do, if you don't mind, is have a round of applause for the panel. This is the refreshing to hear somebody from the federal perspective that has as much passion as you all. This is clearly not the typical bureaucratic program. And I'd also like to thank the Brian and the folks at the caucus for having us here today. For Matt and his team for putting us on. We'll be announcing future events, I think probably next year when we get our breath back again. But also, thank you very much for taking your time to be with us today. We hope you enjoyed it, and have a great morning. Thank you.